So our next speaker is Dr. Michael Segel. He goes by Mickey, right? So, I mean, apropos for Disney World. But he's no Mickey Mouse doctor. Mickey is a Harvard and Columbia trained neurologist who has developed leading models for studying the electrophysiology of nerve cells. He has a special interest in ADHD, attention deficit hypersensitivity disorder, as well as techie bent. He serves on the Electronic Communication Committee of the Child Neurology Society and the Scientific Program Committee of the American Medical Informatics Association. He brings to bear his computer skills in developing diagnostic support software for physicians. He has taken a keen interest in our group, thankfully, and he is one of the few doctors who takes time to interact with us on the PPA forum and former listserv. So please welcome Dr. Segel. Let me stop recording and start recording.
Thank you. And I'm going to be giving two talks, so they're not only somewhat different, but actually almost seem to contradict one another. One of them is about how diagnosis is getting easier and more systematic, and then the other one is about how diagnosis can still be difficult under some situations. And so I'm going to give you examples of both, and by trying to integrate both of those, give you a sense of how far we've come, but how much farther we still have to go. And the initial part about making diagnosis more systematic has been something for which we've received some support from the NIH, both from the National Library of Medicine and from the Genome Institute. And the second is the part that we're doing together with the members of the PPA and Dr. Frank Lehman Horn's group, and that began with our ADHD work and didn't realize how related it was. But my first impression the moment that began was, this reminds me of hypokalemic periodic paralysis, but until members of the PPA told me how much it actually was similar, I didn't really appreciate that. And as you see, those insights have been really crucial in getting to some answers. So I'm going to first begin with the whole situation of diagnosis and discuss why diagnosis isn't simple. I think Jake Levitt gave a pretty good sense of that from all the things that one has to consider, but it's even harder than that in some ways, because when a doctor's trying to evaluate a patient, they're looking at a whole bunch of findings, whether they're physical findings, historical accounts, or lab test results, and then put those together and come up with what the disease is. And it's actually pretty hard, because the way we're taught is if we're reading a review article or a textbook or something like that, or a lecture, it's typically, you're told, here's this disease and here are all the findings of the disease and their various properties. And what a doctor needs to do to make a diagnosis is really to completely invert that. And it's not a simple exercise, and so part of why things go wrong is in terms of diagnosis is that that's hard to do, and people sometimes mess up on that. And so now one of the leading resources for genetic diagnosis has one of the called gene reviews. People would always go to them and say, well, you're telling us about the disease, but we have to do it in the reverse order, so they now link to the diagnostic tool that we've developed to help people do that. And so I'm going to do two things. One is I'm going to show you very briefly using that in terms of the context of doing a clinical diagnosis. But the real focus of the talk, and I'm going to do that only briefly because in a previous meeting we went through that in some detail, and there's also just spare time here and there where I can go through with people individually with that. But what I'm going to show you is some of the new stuff that is happening now. Now that we have access to whole genome analysis, where you can basically look at almost all of the genes in a person and just analyze those together in the context of the clinical situation. And it's amazing new capability that we didn't really have until the last year or two to apply in a clinical context. And it's really going to make medicine very different in lots of good ways, and we hope that we're removing some of the ways in which it's might be scary to physicians and others of overload of information. So the hope of this genome analysis is really that it'll make diagnosis more effective. But the problem was well crystallized in a quote of one of my colleagues, Bruce Korth, who said, we're close to having a $1,000 genome sequence, but this may be accompanied by $1 million interpretation. What I'm going to show is how this can be really taken down to something you can do within seconds. And everything has moved along so dramatically in the last few years that it's something that people who are seeking a diagnosis or trying to help other people get diagnoses should know about. So as with when anything begins, people start with the various fragmented approaches. And so the way people are dealing with interpreting genome information at the moment is both the labs and the clinicians, the physicians, tend to be using these their own fragmented approaches to try to simplify how you deal with the genome. And the reason it's a problem is that when you sequence somebody's genome, you typically get about 30,000 variants that are different from the reference genome. And the question is, well, which of these are important? And so the labs typically filter these 30,000 based on saying which ones look most worrisome from a genetic point of view. But particularly for the periodic paralyses, it's typically just a one base substitution in the DNA, and that might not rise to the top of that. And so the labs are faced with how do you deal with all these huge numbers? If they apply very stringent criteria, they might miss the diagnosis because it doesn't look like a big change in the protein. And they might miss a secondary diagnosis that, as we've discussed here, many people probably have more than one thing going on. And when they've applied looser criteria, they have huge amounts of genes to look at, and the analyses can take several weeks of physician time and therefore cost a lot of money, and that's what gave rise to the concern about the $1 million interpretation. And the clinicians have been looking at this in a different way, that they've been saying, well, I can say that this is in this group of diseases, so let's just look for this group of diseases. And you could have a periodic paralysis panel, or you could have a channelopathy panel, but then there'll be other things that sort of look like it that aren't in that. And so if you take that sort of narrow view, you're potentially missing other things also. And so what we've been doing has been to take an integrated approach of looking at everything and just giving everything scores. So we look at the clinical situation, and we come up with likely diseases based on the clinical situation, as we've been doing for several years, and I've shown in a previous meeting, and I'll show briefly here. But then we also do the same thing for all these 30,000 or so genetic variants, and then put them together and see which ones could fit together and produce particular disease patterns and so forth. And the idea is that if you do this complete analysis of all the clinical information versus all the diseases and all the genes and so forth, then you're really getting the most complete look at the situation. Now, of course, it sounds like an immense amount of work to do, but computers are fast, and the whole thing takes a second or two. So it's really remarkable what all this knowledge about genes is allowing us to do. So I think what I will do now is, oops,
Don't click that X.
am I okay?
Yeah, just don't click the X, that's all.
Okay, I have to click this. So I've get rid of the advanced mode so everything looks simple. Try not to click that X. So what I'm going to do briefly is just show you how the basic diagnostic software works by putting in a very simple scenario of a periodic paralysis type of situation. And it's going to start out with a very broad differential diagnosis because there's lots of things. So we'll put in intermittent or recurrent episodes of weakness, and that can be because of your joints, it can be because of all sorts of things. But let me just do that to get going in a very general context and then show you how we can help narrow things down. So we'll put in a 25-year-old man, and we have some quick lists of commonly used findings for different fields, and so we can say that there's so let's say there was weakness that was present, and we often in clinical medicine the age at which things happened is important. You can't just say present or absent. And so let's say the weakness had onset at 15 years old, and there were recurrent exacerbations with onset at about 15 years old. So like you had your first one around 10 years old and then another one. So this is very, very general information. And we'll go to a differential diagnosis and we'll get a very general thing that will include everything from arthritis to things like narcolepsy and so forth. And a lot of the things that have been considered for various of you, mitochondrial diseases, myasthenia, and so forth will appear here, but also, of course, hypokalemic periodic paralysis. Now, a lot of these diseases are showing up very higher than periodic paralysis because we take incidence into account by default, and if we take that off, hypokalemic periodic paralysis comes to the top. But we wouldn't expect that. We would expect people to take the, let's not focus on the zebras at the beginning. And then we also encourage people to add more findings. And so so first of all, the patient tab is what we already put in. And the add findings looks at all the other possible findings in the database, of which there are a total of 7,000 that include mostly lab tests, but also a lot of clinical information, and asks various questions, such as are there prominent headaches, or is there intellectual disability? And you can say that's absent. Is it worse following activity? You can say yes, that's present. And if you note on the side, some of the diseases are starting to change their order. So hypokalemic periodic paralysis is rising up. And so we can try to get rid of some of the arthritis type of things. And we can recalculate this for the new differential diagnosis, and we'll start focusing on some of the things like let's get rid of the foot problem, and so forth. And so you could imagine that somebody would go through this analysis and they'd be thinking of mitochondrial diseases, myasthenia gravis, and hypokalemic periodic paralysis. And this is a better situation than somebody saying, I know this must be myasthenia gravis and I'm going to treat this way. And so this is, and then when people would go to, let's get rid of one more of these things. Well, let's just recalculate, and we're starting to see, I guess if we go a little lower, you get things. It starts asking if having sodium triggers attacks, or carbohydrates make it worse, or potassium makes it better, and stuff like that. So you see that there's very much of a focus on the clinical approach here, and that by going through these types of things and saying there's no joint pain and so forth, you can very quickly, from a clinical point of view, start converging on the likely things. So we can go through more types of specific situations later. What I want to do is switch to another type of muscle disease patient, one for whom we have a full genome, and it was a very difficult case, but it shows you how by applying the genome you can reach diagnoses that you just wouldn't have been able to reach before. So I'm going to load a file with the patient's findings. And what this patient has is ptosis of the eyes, which is a sort of droopiness of the eyes that's often seen in congenital myasthenia. They have motor developmental delay, EMG changes, muscle biopsy abnormalities, and deafness. And the drugs that are used to treat congenital myasthenia improve the condition in this patient. So this is a diagnostic dilemma. And it's a diagnostic dilemma because you could say, okay, well, maybe this is a muscle disease, but why is there deafness? So you could say, well, there are mitochondrial diseases that have deafness plus muscle weakness, but on the other hand, the congenital, the ptosis and the cholinesterase inhibitors working suggests that maybe this is myasthenia, this is congenital myasthenia, but that doesn't have deafness. And so it's sort of a puzzle, but it gets cleared up a lot when you import the genome, which I will do by importing what's called a genome variant file. This has about 30,000 variants. And as you'll see, this all happens virtually instantly, and you get a report of 32,000 variants, and reports various quality metrics so that help people decide whether there's any problems with the samples and so forth. But then when you go back to the differential diagnosis, the patient tab is now replaced by a phenotype tab, which is a description of the clinical findings, and a genotype tab, which describes the relevant genes. And one is able to attach a pertinence measure to genes that pull out which of the genes are most important in this patient. And for an individual gene, you can click on it and see all the variants that were found in this patient, and take a look at the details of them and so forth, but I won't dwell on that. But the point is here that this patient turned out to have two things. They turned out to have a mutation in the Titin gene, that's the TTN, that causes Segal myopathy. And the severity of the mutations was given a score of 2 out of 4. And the C indicates a compound heterozygote, meaning that they got one. There's an abnormality in each copy, and this is a recessive disease, and they got one from each parent. And similarly, but if you look at the details of Segal myopathy, it doesn't include deafness, but you may notice down here there's GJB2 related deafness, and the number 2 gene in terms of the gene pertinence is the GJB2 gene mutations. So what this analysis does is, can't let this go wrong. What this analysis does is it allows a doctor in a completely hypothesis-independent way to just put in the observable things about a patient and then import the genome completely hypothesis dependent, doesn't assume it's recessive, doesn't assume it's dominant, and so forth, doesn't assume it's one gene or two genes, and pulls out the relevant details in a process that basically just takes seconds. So this is sort of this is where medicine is going in terms of the use of technology. In some ways it's more clinical because we're focusing very much on the clinical findings, and some of the doctors have said, well, I'm going to be using this, I'll use this stuff with a patient in the room, because who would have thought about asking about whether carbohydrates make them worse if I wasn't even thinking of periodic paralysis, and there's about 2,700 and something genes for which we know of a clinical disease attached to them. And I can assure you that no doctor is, even those of us who've actually gone through each of these genes, I can assure you we don't remember all the details of them. And the idea is to make this information available to the doctor in a just-in-time basis in a way that is very clinical, so they can ask you, did it do this and that, and so forth. And at a previous session, we actually went into the database and added a finding or two to hypokalemic periodic paralysis, because even the literature won't have some of the things that those of you who have proven mutations can say, we have that, I don't know why it's never been documented, and we could just put it in. So you have the ability to collect the clinical information and go from that directly to the genetic information. And so this is what will make the power of this whole genome analysis something that can be used on a relatively common basis. And when the costs get down to $1,000 or a few $1,000s, it starts to be comparable to some of the other types of tests we're discussing, such as muscle MRI or EMG or muscle biopsy and so forth. And I think what you'll be seeing in medicine is lots more application of genome analysis, and there's about 22,000 or so genes in total. We know only about 15% of them in terms of their clinical patterns. That information is coming out at a huge rate now, and the second talk that I give will give you a sense of how that's going to impact periodic paralysis. So the whole ability to do this type of analysis is going to be a core part of medical practice, certainly for specialists. I don't know how much it'll be used by generalists, but for a specialist to query everything that's known about genetic disease, this is the wave of the future. And so I think I'll pause here. If there's questions about this part, we can get into the questions now. And then afterwards, I'll do the other part of the talk, which is about the lidocaine insensitive form of periodic paralysis.
Oh, yeah, sorry, yeah, we gotta do the...
Oh, so are there two microphones?
No, unfortunately, this is the only one that works there, so...
Oh, so I need to come, because I'll...
Yeah.
So thank you. It's a great tool. How can physicians get access to that?
Physicians can use the general diagnostic tool for, they can use that for free. The other is made available through labs that pay a fee to use it. And part of the deal with the NIH is that we're making this free for everybody to use in clinical diagnosis. And there's thousands of users in 100 countries and so forth.
So you can use it in Europe as well?
Absolutely. I mean, there's 100 countries. It's hard to get to 100 countries without getting to Europe.
Just one quick question. The VA did a DNA test on me. It came back inconclusive. Is that very well standardized? Or is there different labs doing it different ways?
Well, what I was showing here was something that's usually termed whole exome or whole genome studies, which are still relatively unusual. And typically what's done is sequencing that just checks a particular gene of interest or a particular part of a gene of interest. So when you send your blood to Dr. Lehman Horn, typically he's doing that simple type of test. When, as you'll see in the second part of my talk, when you were trying to find a new gene, then you start doing things like looking at the whole thing. And so he is doing that with some of them, and there's other techniques that have whole collections of genes, but not everything. And there's lots happening. It's one of the really fast-moving areas of technology. It's sort of like the early 1980s in terms of personal computers. It's stuff that we know how to do at big, expensive scale, but it's coming right down to the personal level.
I noticed, you pulled up narcolepsy was high on the list, and I used to be one of those people. When I was young, I could fall asleep anywhere. Now, if I don't have my Adderall, I'd be crashed out through this whole meeting.
Can you repeat the comment?
No, I'm
Sorry.
No, I was just saying that I noticed in the list that narcolepsy and ADHD were high up there. My son's been diagnosed, and the VA doesn't recognize it because I'm over a certain age. But if I'm not moving or taking Adderall, I would fall asleep in the middle of a conversation at the position of attention in the Army. So untreated, if I wasn't moving, I was asleep. And now I'm noticing that it's all, every time I turn around, I see the word narcolepsy and ADHD, and it makes me think that there's a bunch of links there.
Well, what's really important is often there are a bunch of diseases that look similar, but have very different causes. And it's really important to think of all of them because you don't want to get the wrong one. And specifically, there's a great example with hypokalemic periodic paralysis that you can cause the same thing in ways that are pretty much indistinguishable clinically with a sodium channel mutation or a calcium channel mutation causes the same thing. And what they're both doing is sort of overloading the cell and running down its energy, and then you can't, even if it was a calcium coming in, and you run down the energy so much that sodium starts coming in because you're running out of energy. And so there are ways where often you'll see things that travel together in terms of having similar clinical findings, but when you get down to the molecular mechanism, they're different, but they feed into the same final common pathway. And then there's other things like narcolepsy where we don't really have a very good understanding of what causes that. We just know that it exists. But what's important is that somebody should be able to focus on, they should be able to consider those, and without providing a strong reason why it's one thing and not another, narcolepsy should still be up there and people should be considering it. And if we had focused on it specifically, there were questions there that were specifically asking about narcolepsy. Do you tend to fall asleep during the day and stuff like that? And so it's really important not to miss any of these things. So that the purpose of the PPA is not just to diagnose periodic paralyses, but also to help you if you have something else that might look like this. And similarly, that's the attitude of doctors. It's just that doing this whole process in your head is so cognitively difficult, and it's hard to remember all these things that people often use shortcuts, and shortcuts sometimes miss things.
Vicki, I think I wonder, are there like a lot of questions? One? Maybe we'll do one more question and then have a break for 10 minutes, because I think it's time for a break. And then we'll start again. So one more question. Okay.
You said about deafness. Are you saying that gene has something to do with hypokalemic periodic paralysis? Because I have all deaf people in my family, so I'm just wondering, or did I misunderstand it?
So I had done a quick switcheroo and said, let's start with a different patient. And I put up this patient with muscle weakness and deafness. And what we were able to show here was that the muscle weakness and the deafness were due to two different genes. And we were able to pull those two genes out of a whole 20-something thousand genes and pull them out such that they came up number one and number two in the analysis. And so in that case, it was two separate genes. And sometimes that's the answer. And in fact, as I had mentioned before, it used to be that we assumed that that adults who were old had a bunch of chronic diseases, and they had several things going on, but kids had only one. But here was an example of a kid who had two things going on, and increasingly, as people are looking, they are finding that there's many situations where people have two things going on. And I'll get into this theme a little more in the other talk, because I think there's examples where somebody has a gene, but then there's another gene that modifies how severely the main gene is expressed.
Great, thanks. Okay, so we'll take, let's take a break and be back at 3:25.
Everybody, people told me to hold the microphone closer, so this time you should complain if it's too close. So before I try to give you a sense of how diagnosis is getting significantly easier, both in terms of automated access to information and automated analysis of your entire genome of 22,000 genes. But the theme of the second part is why diagnosis is still difficult, and why we only know about 2,700 out of the 22,000 genes. And sort of we're like 15% of the way there in terms of the diseases. We won't have a disease for every gene, because there's some are redundant, or some you die before you even get born, and so forth. But anyway, there's a lot to learn, and I'm going to give you an example that is very close to the interests of many of you in terms of periodic paralysis. For the molecular diagnosis of hypokalemic periodic paralysis, some people will oversimplify this to you and say that there's only two genes, and if you don't have one of those two genes, you don't have hypokalemic periodic paralysis. That's a vast oversimplification. The numbers as we have them are 60% of people who clinically look like they have hypokalemic periodic paralysis have a mutation in the calcium channel known as CACN, which stands for calcium channel A1S. And about 10% have a mutation in a sodium channel. That's what the SCN part is, number 4A. But those only add up to 70%. There's a bunch of people who don't have a diagnosis. And several years ago, it became clear that among the people who didn't have a known gene, there was a common clinical pattern, which I'll describe in some detail of what we'll call here the lidocaine-insensitive type of hypokalemic periodic paralysis. And when I say lidocaine-insensitive, it means lidocaine that's often used as a numbing medicine doesn't work well for these people. And 2 years ago at the meeting, we went around putting dabs of lidocaine gel on people's tongue, and a lot of people said, yeah, this works just fine for me, and a whole bunch of people said, doesn't seem to do anything to me. And that's what we mean by lidocaine insensitive. And about 4% of the entire population is lidocaine insensitive, so we don't think that all of them have hypokalemic periodic paralysis, but we actually do think that a lot of them do have ADHD, and I'll get into the significance of that. it's likely that even beyond these three genes, so we think there's a third gene, and I'll give you the update on how Dr. Lehman-Horn's group is converging on identifying that gene. There could easily be other genes. We shouldn't say we're stopping at 3, just like other people shouldn't say they're stopping at 2. Typically, the history of these is you keep finding genes for typically smaller groups, but here I think that it's possible this gene is going to be above the 10% for the sodium channel. There's also other diseases that clinically look like hypokalemic periodic paralysis that aren't. And many of those have been discussed in the various presentations. Some of those you saw come up on the differential diagnosis when I went through with the diagnostic software. And depending on the specific clinical findings that you put in to software or you're thinking about, different of these diseases will be relevant. And so there's really a whole process to trying to figure out what somebody has. My guess is of the people in the room here, of the symptomatic people, not everybody actually has a periodic paralysis, although some of them have some of these other periodic paralyses that are on the list. Some of these have other diseases that are similar to periodic paralysis in their clinical appearance. So the lidocaine-insensitive type of hypokalemic periodic paralysis, it looks, one of the other names that we've used for this is hypokalemic periodic paralysis plus, because it has all the features of hypokalemic periodic paralysis, which I've listed here as the episodic weakness and the various triggers that have been discussed, and that including low potassium, but also the potassium goes even lower during the episodes. All of these are present. But in addition, there's 3 other interesting things. One is that, as I mentioned, lidocaine hardly works as a local anesthetic. So the typical history you get is I go to the dentist, they have to give me 3 shots of lidocaine, and it takes 45 minutes before it takes enough effect that I can have dental work, or that it doesn't ever take effect and they do the dental work anyway and they diagnose me as having dental anxiety. So, those of you who have been accused of having hysterical paralysis, this is sort of like minor compared to the other things you do, but this probably happens quite a bit, as I'll describe. The other thing is that a lot of these people, and particularly the males, often get the diagnosis of either ADHD, which is attention deficit hyperactivity disorder, or ADD, which is just the same without the hyperactivity part, and they're probably it's probably the same genes, and I wouldn't really distinguish among them. Or Asperger's syndrome, which is classified, the psychiatrist decided to move that into what they call the autism spectrum, but it's really clinically indistinguishable from ADHD, and I think they made a really big mistake. And when we find the genes for these things, they're going to be very embarrassed, but we warned them in advance, and they did it anyway. So it's their problem, not our problem. But the other weird thing is that the females who have this tend to have severe PMS, premenstrual syndrome. And you might think, what does premenstrual syndrome have to do with ADHD? It's sort of like you wouldn't think of them as in the same category at all. But if you actually think about it, both are situations in which you are being bombarded with sensory input that you can't turn off and it's bothering you. One of my favorite descriptions of ADHD was in a video that the New York Times had on their website of some guy who described ADHD as being like in a room with a whole wall full of TV sets, and they're all on to different channels, and you don't have the remote. And that's what it's like. It's the inability to focus in on one thing that many of the rest of us just find sort of natural. We can be in a cocktail party and pay attention to one person. These people are paying attention to everything. And in the old days, thousands of years ago, if you were out hunting, that's exactly what you wanted. You wanted to pay attention to this sound there or that sound. It could be what you were looking to hunt or what might be out there hunting you. And this was probably what one wanted 10,000 years ago. It's not necessarily what one wants in today's world, and it's not really clear, or it's certainly possible that some of what we call ADHD today was actually the ancestral state, and what we're and those of us who don't have ADHD are mutants in one way or another. And so it really sort of turns these things on their head. But anyway, so this is a combination of things that people have, and we include here in the criteria no known mutations in the calcium channel or the sodium channel, because about 4% of the population has this combination of lidocaine insensitivity and ADHD. We think that on average 4% of people with hypokalemic periodic paralysis due to a sodium or calcium channel mutation are also going to be lidocaine sensitive just by coincidence, because you'd expect 4% of any group of people to have those. And the inheritance is autosomal dominant, which is the same as it is for the other channelopathies, which means that if you have it, the chances are pretty good you got it from one of your parents who's also affected, and so forth, and that on average half of your kids would have it, and so forth. And we've been able to follow this through some very large families, and that's the basis for the work that Dr. Lehman-Horn's group is doing, is following this through some very large families, and I'll talk about that. But just as a background for here, you've heard me talk about how this is, we think this is common. The way we got involved with this, and my background is with ion channels and epilepsy, and I stumbled across situations in which people had the combination of the lidocaine effect, the ADHD, or the PMS in their families, and I thought, wow, this is amazing. We're able to treat these people with potassium as if they have hypokalemic periodic paralysis. And it was only when I was contacted by some PPA members who said, well, there's actually is a form of hypokalemic periodic paralysis. There's a bunch of us who have this in addition, that we were able to say, well, this might be an even better model for looking at the genes, because it's much easier to trace periodic paralysis through a big family than it is to trace ADHD, particularly since many females are, 75% of females who have ADHD don't get diagnosed as having ADHD. Just they're able to keep the lid on better than guys can for some reason that nobody quite understands. So Dr. Lehman-Horn's group is working on finding the gene. Most of you who are submitting blood samples are just doing some of the standard testing for the 2 known genes. But some of the PPA families who have this lidocaine insensitive form, and particularly have large families where you can trace where you can trace the gene for many families, are involved in the early effort to find the gene. But then once that gene is found, then they will be testing for 3 genes instead of 2 genes. And so this is how the science progresses, beginning where it's easiest with the big families. And to explain to you how those studies, let me give you a sense of the basic technique that's being used. So first of all, on some of these, they've tried whole exome sequencing, and the problem with whole exome sequencing is the exome is only the parts of a gene that are translated into proteins. It doesn't include the part between the exons within a gene that's called the introns. It doesn't include the other 98% of the DNA that's between genes, and that's involved. And it sometimes doesn't even include the first exon out of exons in a gene, just because the techniques don't sequence them very well. So they are using a more standard technique of trying to find new genes, and I think the best way of visualizing how that works is you can study a lot of single DNA-based changes that just happen to be very different among different people, just random, and they're not associated with diseases, but they just provide a way of sort of labeling it. And so there's four different DNA bases it can be at one point. So if you think of 4 different colors, you can go through like 100 places on everybody's chromosome and just put a band of color based on which DNA base they have. So if you can imagine that it's kind of like painting the chromosomes so they have a particular pattern. And then what you do is you look in a big family and you look, hey, these 2 second cousins have it. And let's check, and your various chromosomes you get from different ancestors and so forth, and this allows you to go trace through which you got that's in common. And so you can very quickly localize down, especially if you have a very large family and distant relatives who have the same thing, you can zero in on a particular region. So that's how one finds a region, and but once one finds the region, one's not always you don't always have the answer the next day, and there's various reasons why this can be the case. The gene can be in the region, but it can be not part of the 1% of DNA that's known to be genes. The gene could be one that's already known, but as I mentioned in whole exome sequencing, that you can't be guaranteed that all the parts, all the exomes that make up a gene, you can't be guaranteed that each of those is sequenced well. The abnormality can be within one of these introns between the exons in a gene, or the abnormality can be in regulatory DNA outside a gene. So for instance, you can have a sequence outside a gene that controls the amount of something like a potassium channel, and if you're producing too little of a potassium channel, and even if you have regular sodium and calcium channels, you could have the amount of current coming in through your channels can be greater. So there's still lots of possibilities. There's some meticulous work to get to this point, but we think it's not only important to be able to knock off another 10 or 15% and say these people have this gene, but there's potential applicability to understanding the molecular mechanism of ADHD. So there's a lot of interest in terms of solving this problem. And from the point of view of people with periodic paralyses, it's the number of people who have ADHD is probably about 1,000 times as great as the number of people who have hypokalemic periodic paralysis. So one of the best things that could happen to the Periodic Paralysis Association is to have another similar channelopathy that's 1,000 times more common, because that brings in a huge amount of interest in terms of people developing new therapies, in terms of just general awareness of that this is one of these tip of the iceberg diseases, and all doctors will know about this because this will be how you learn about a common disease. So it's potentially of big interest both to the periodic paralysis community, but also to the wider community, and it's been terrific for a lot of people who have ADHD, who have this lidocaine-insensitive form, that they've been able to go on to periodic paralysis type of treatments with potassium and so forth, and avoiding sodium. And people's response typically is, I had no idea the answer could be anything this simple. So that's what I prepared. The the sort of double message of how diagnosis in general is getting easier and more systematic, but how there's still stuff to discover, but once we discover it, it's going to be part of the stuff that's easier and more systematic, and we still have a long way to go, and part of the way we do it is through the insights that where you guys can help those of us who are trying to do the research, and most importantly, the ability to work with some of the families, and particularly for this, some of the large families, to be able to pull out an entirely new gene that's going to have a big effect on the periodic paralysis community, but also potentially much wider as well.